Hello learners, we have seen in our earlier modules the chemistry, structure, bioavailability and metabolism of curcumin. Let's now learn the very important aspect of curcumin, that its impact on health. Curcumin is a major source of polyphenol. It aids in management of oxidative and inflammatory condition, metabolic syndrome, arthritis, anxiety and hyperlipidemia. It may also help in management of exercise-induced inflammation and muscle soreness, thus enhancing recovery and performance in active people. In addition, a relatively low dose of complex can provide a health benefits for the people that do not have diagnosed any health condition. Most of these benefits can be attributed to its antioxidant and anti-inflammatory effect. Let's now understand the basic mechanism involved in imparting the health benefits of curcumin. Let us now learn the health benefits of curcumin. Antioxidant effect. Antioxidant and anti-inflammatory properties are the two primary mechanisms that explain the majority effect of curcumin on various conditions. Curcumin has been shown to improve systemic marker of oxidative stress. There is evidence that it can increase serum activities of antioxidant such as superoxide dismutase. In recent systemic review and meta-analysis of randomized control data related to efficacy of supplementation with purified curcuminoids on oxidative stress parameters indicated a significant effect of curcuminoid supplementation on all investigated parameters of oxidative stress, including plasma activities of superoxide dismutase and catalase, as well as serum concentration of glutathione peroxidase and lipid peroxides. It is noteworthy to point out that all the studies included in the meta-analysis utilized some sort of formulation to overcome the bioavailability challenges, and 4 out of 6 used piperine. Curcumin effect on free radical is carried out by several different mechanisms. It can scavenge different form of free radicals such as reactive oxygen and nitrogen species. Superoxide dismutate enzymes active in neutralization of free radicals. Also, it can inhibit ROS generating enzymes such as lipooxygenase, cyclooxygenase and xanthine hydroxygenase and oxidase. In addition, curcumin is a lipophilic compound which makes it an efficient scavenger of peroxyl radicals. Therefore, like vitamin E, curcumin also considered as chain-breaking antioxidant, anti-inflammatory. Oxidative stress has been implicated in many chronic diseases, and its pathological process are closely related to those of inflammation, in that one can easily induced by another. Tumor necrosis factor is a major mediator of inflammation in most diseases, and this effect is regulated by activation of transcription factor, nuclear factor. In addition, tumor necrosis factor alpha necrosis factor is also activated by most inflammatory cytokines. Gram-negative bacteria, various diseases causing viruses, environmental pollutants, chemical, physical, mechanical and psychological stress, high glucose, fatty acids, ultraviolet radiation, cigarette smoke, and other disease-causing factors. Curcumin has been shown to block nuclear factor activation increased by several different inflammatory stimuli. Let us now learn effect of curcumin on metabolic syndrome. Metabolic syndrome induces a cluster of conditions like insulin resistance, hyperglycemia, hypertension, low density lipoprotein cholesterol, elevated low density lipoprotein cholesterol, especially the visceral obesity. There is evidence that curcuminoids modulate the expression of genes and the activity of enzymes involved in lipoprotein metabolism that lead to reduction in plasma triglycerides and cholesterol and elevate high density lipoprotein cholesterol concentration. In randomized double-blind placebo-controlled trial with parallel group design, 117 subjects with metabolic syndrome received either 1 gram of curcumin plus 10 mg of piperine to increase the absorption for 8 weeks. Within the group, the analysis revealed significant reduction in serum concentration of tumor necrosis factor, transforming growth factor beta TGF, and monocytes chemoattractant protein 1 MCP following curcumin supplementation. The result of this study suggests that curcumin supplementation significantly decreased serum concentration of pro-inflammatory cytokines in subjects with a metabolic syndrome. Study also concluded that reduction in total cholesterol, reduced serum triglycerides, and improvement in HDL cholesterol levels. Let us now learn the effect of curcumin on cancer. In normal cells, there is a finely controlled balance between growth promoting and growth restaining signals such as proliferation occurs only when required. Example, during wound healing and during normal tissue turnover. Proliferation and differentiation of cells during these processes occurred in ordered manner and cases were no longer required. In other words, normal cell becomes malignant when cellular proliferation is no longer under normal growth control. There are, of course, other characteristics that cancer cell may possess, such as angiogenesis, metastasis, and suppression of apoptosis. But at the end, the uncontrolled proliferation of the cell is at the heart of the disease. Therefore, to understand the cancer, we need to transfer our knowledge on cell proliferation and its control. Among these phytochemicals, curcumin has been identified as one of the major neutral anti-cancer agent exerting anti-neoplastic activity in various types of cancer cell. It is hypothesized that curcumin assert its anti-tumor activity in cancer cell by altering deregulated cell cycle via cyclin dependent, P3 dependent, P53 dependent, P53 independent pathway. The anti-tumor effect of curcumin has also been attributed in a part to suppress of cell proliferation, reduction of tumor load, and induction of apoptosis in various cancer models in both in vitro and in vivo. It induces P53 gene dependent apoptosis in various cancer of colon, breast, bladder, neuron, lung, ovary, etc. Although both p53 dependent and independent G2 M phase arrest by curcumin has been observed in colorectal cancer cell. Curcumin inhibits angiogenesis directly via regulation of angiogenic growth factor like vascular endothelial growth factor, basic fibroblast growth factor and epidermal growth factor, as well as the genes like angiopoietin and hypoxia-inducible factor, heme oxygenase and transcriptional factor like nuclear factor. Curcumin can act as a potent immunomodulatory agent that can modulate the activation of T cells, beta cells, macrophages, neutrophils, natural killer cells and dendritic cells. Few research have also demonstrated that chemopreventive role of curcumin. The dose of curcumin depends and decided by type of cancer, its metastasis condition. Learners, let's now learn about wound healing properties of curcumin. Tissue repair and wound healing are complex processes that involve inflammation, granulation and tissue remodeling. Interaction of different cell, extracellular matrix protein and their receptors are involved in wound healing and are mediated by cytokines and growth factor. Curcumin enhances cutaneous wound healing by increasing the formation of granulation of tissue as reported by studies done on animal like rats. Curcumin has also shown its promising result in control of arthritis, control of infection and improved liver function. Let's conclude, learner. Turmeric has been used for thousands of years in Ayurvedic and traditional Chinese medicine. In modern time, curcumin, the yellow pigment of the spice turmeric, continues to be used as alternative medicinal agent in many parts of Southeast Asia for treating common health problems such as stomach upset, flatulence, jaundice, arthritis, wound and skin infection among many others. Curcumin and turmeric products have been characterized as safe by health authorities such as FDA in United States of America and FAO as well as WHO organizations.